This is a summary of an article that the Alzstatin project at AlzeCure Ferm just recently published. And this is the article published in Journal of Pharmacology and Experimental Therapeutics entitled Gamma-secretase modulation inhibits amyloid plaque formation and growth and stimulates plaque regression in amyloid precursor protein pre-signaling 1 mice. And this is a collaboration between a number of groups from both Karolinska Institute, Washington University, and Gothenburg University. And the aim of the study was to investigate biological mechanisms relating to gamma-secretase modulators as novel therapeutics for the treatments of Alzheimer's disease. A brief background on the amyloid cascade in Alzheimer's disease. The protein APP in the plasma membrane is cleaved first by beta-secretase base and then by gamma-secretase to generate A-beta of varying length. The short forms of A-beta, A-beta 37 and 38, are more soluble forms, whereas the longer forms, such as A-beta 42, are insoluble and can easily aggregate into plaques. Aggregated forms of A-beta have been shown to be toxic and can subsequently be an initiator of tau pathology and then also to impaired cognition. Recent data from studies with A-beta antibodies have shown that removing the plaques has beneficial effects on the progression of disease. Thus, reducing the production of A-beta 42 will stop this cascade of events. Briefly about the mechanism of gamma-secretase modulators, GSMs. After base cleavage, the gamma-secretase cleaves the remaining part of APP by several subsequent cleavages into mainly A-beta 40 and 42. What a GSM does is to stabilize the gamma-secretase complex such that more cleavages can be performed, allowing more shorter forms of A-beta to be generated. Thus, less of the longer forms of A-beta will be produced and more of the shorter forms. Gamma-secretase inhibitors have been extensively studied in Alzheimer's disease, but have been shown to have serious side effects. However, there is a clear differentiation between a gamma-secretase modulator and a gamma-secretase inhibitor. An inhibitor inhibits all gamma-secretase mediated cleavage and will lead to the accumulation of the substrate in the plasma membrane and the inhibition of any intracellular signaling of the C-terminal domain. This is a problem since gamma-secretase has more than 100 substrates. Such an inhibition will affect many cell functions, one of which is the inhibition of the notch cleavage, which will stop the important notch signaling pathway. Gamma-secretase modulators, on the other hand, do not block gamma-secretase. Thus, substrate is not accumulated and intracellular signaling is not affected. Therefore, we believe that gamma-secretase modulators are a selective and a safe way to reduce the levels of A-beta 42. GSMs produce more of the shorter peptides, A-beta 37 and 38, and less of the longer forms. And several studies have shown beneficial effects of these short forms of A-beta. There is clinical evidence that in a population with A-beta pathology, but with high A-beta 38 levels, there is a slower decline in memory as measured by MMSE and a slower conversion from mild cognitive impairment to Alzheimer's disease. There is also preclinical evidence that A-beta 38 can attenuate the negative effects of A-beta 42 on long-term potentiation, which is an essential part in memory formation. And as well as that the shorter forms of A beta can attenuate A beta 42 mediated toxicity. In the paper, we have studied the interaction between different A beta species and how they interact in the form of aggregation. In an aggregation assay in vitro, we added A beta 42 and then the other peptides, A beta 37, 38, 40, and 42, to that basal level of A beta 42. And then we saw a decreased aggregation when we added A beta 37 and 38 to the available A beta 42, but we saw more aggregation when we added 40 or 42. And in a second experiment, we also studied different concentrations of A beta 37 added to A beta 42. So the conclusion is that the shorter peptides, both A beta 37 and 38, reduces the aggregation of A beta 42. In the remainder of the paper, we used a tool compound, a potent GSM entitled AZ4126, which originates from AstraZeneca. It's a well-characterized GSM. We studied it in a cell system containing human inducible pluripotent stem cell-derived cortical neurons. We measured the levels of A beta 42, 40, 38, and 37, and we saw the expected decrease both in 42 and 40, as well as an increase in the shorter peptides 37 and 38. Then we studied the compound in vivo microdialysis in TG2576 mice, a transgenic mouse model of Alzheimer's disease. We used both 6-month-old and 15-month-old animals. We saw a rapid, large, and sustained reduction of both A beta 42 and 40. But we also saw, as depicted on the graph in the bottom, a large increase in A beta 37. We then used 2-photon microscopy in order to study individual plaques in vivo and how they are affected after a GSM treatment. We used the 6-month-old APP PS1 transgenic mice and treated them for 28 days orally with the GSM AZ4126. In this model, the skull bone of the mice is thinned, which allows us to use 2-photon microscopy to visualize individual A beta plaques in the brain that are labeled with the compound methoxyXO4. And here are the results from this study. On the left, we can see animals treated with vehicle. And after 28 days, we can see a number of plaques that have grown in size, indicated by green arrows. And also we see the birth of new plaques indicated by blue arrows. When the animals were treated with the GSM AZ4126, we saw that a lot of plaques that were unchanged in size, indicated by yellow arrows, but also plaques that have had a reduction in size after the 28-day treatment. That is indicated by the red arrows. Compiling these data demonstrates that, as indicated in the figure C, the GSM reduced plaque growth, reduced the appearance of plaques as seen in figure D. Figure E shows that 30% of the plaques have a reduction in size after the 28-day treatment period with the GSM. Thus, a GSM appeared to be able to change the plaque dynamics from growth to regression. Interestingly, this has not been observed with either BACE inhibitors or gamma-secretase inhibitors. Our hypothesis is that it is the increase in the short form of A-beta 37, that contributes to the plaque regression. In summary, gamma-secretase modulation, which does not block the enzyme activity, provides a safe way to reduce the amount of aggregation-prone A-beta 42 as well as 40, and also it will increase the levels of A-beta 37 and 38. We have shown that these shorter A-beta peptides, 37, 38, can inhibit the aggregation of A-beta 42. We also have seen that the tool compound, the GSM AZ4126, produces a very characteristic A-beta peptide pattern of a GSM, both in a translational cell system as well as in vivo in a mouse model of Alzheimer's disease. We've shown that the compound, the GSM, can reduce amyloid plaque size in a mouse model of Alzheimer's disease after a 1-month treatment. Therefore, we believe that GSM is a very attractive therapy for the therapeutic use in Alzheimer's disease.